Welcome to the Deep Dive, the show where we unlock the most important nuggets of knowledge from a mountain of information, turning complex topics into clear, surprising insights. Today, we're embarking on a truly fundamental journey right into the very core of how your body works. Have you ever wondered what's truly happening inside you when, say, a sudden noise makes your heart race, or when that persistent hunger directs your search for food? Behind every single one of those seemingly simple everyday experiences, behind every coordinated response your body mounts, lies an astonishingly intricate, highly coordinated system of cellular communication. Okay, let's unpack this. Today, our deep dive is into the incredible world of hormones. We're not just going to skim the surface of what they are, we're going to dive deep into how they actually work inside your cells. We're talking about the precise molecular choreography of how your body takes an external cue, or maybe an internal need, and translates it into a specific, targeted action at the cellular level. Our mission for you, our dedicated listener, is not just to grasp the basics, but to truly achieve a mastery-level understanding of hormone action and signal transduction. The kind of nuanced insight that, well, the elite professionals in this field possess. Because when you truly master these fundamental mechanisms, you'll discover how these intricate pathways are often at the very heart of disease, and crucially, how they become the primary targets for powerful, life-changing medicines that shape modern healthcare.
And to guide us on this deep dive, we're drawing from an exceptionally rich and detailed source. It's a comprehensive look into the biomedical importance of hormones, the countless, frankly ingenious ways they send signals throughout the body, and the intricate downstream effects that ultimately shape our biology, our health, and really our very existence. So prepare to see the inner workings of your body with a clarity and depth you might not have imagined possible.
Let's begin by setting the stage, what we're calling the grand orchestration, hormones and homeostasis. At its most fundamental level, the biomedical importance of hormones comes down to one central, overarching idea. They are the major facilitators of homeostatic adaptations. Think of your body as a constantly adjusting, self-regulating marvel, always striving for balance amidst a constantly changing environment, both inside and out. Hormones are, well, the maestros conducting this adaptive symphony.
That's a perfect way to put it. Their core function is astonishingly precise. They alter the activity and the sheer amount of proteins within cells. It's not simply a matter of flipping a switch on or off. It's about fine-tuning the levels and the precise actions of the molecular machinery inside every cell. And here's a profound insight, really, that sets the stage for everything we'll discuss: the concept of signal amplification. Imagine a whisper triggering a roar. A seemingly tiny interaction, perhaps just a single hormone molecule binding to its receptor, can generate an incredibly amplified intracellular signal. This means even a minute amount of hormone can create a widespread, powerful cellular response.
That amplification is truly remarkable because it means that even, subtle fluctuations in hormone levels can have widespread cascading effects throughout the body. These signals can directly regulate specific genes, which, in turn, changes the amount of proteins being produced by a cell. But it's more immediate than that, too. They can directly affect the activity of existing proteins, like turning enzymes on or off, modulating transporters that move substances across membranes, or opening and closing channel proteins that allow ions to flow. This can instantly change how a cell behaves. They can even influence the precise location of proteins within the cell, directing them to exactly where they're needed to carry out a particular function. And on a broader scale, these hormonal signals impact fundamental cellular processes like protein synthesis, guiding the creation of new proteins, cell growth, and even cell replication, all through their carefully orchestrated effects on gene expression and protein activity.
It's also vital to grasp that this intricate work isn't confined to what we traditionally think of as hormones. Other crucial signaling molecule substances like cytokines, which are key communicators in the immune system, interleukins, which mediate communication between white blood cells, growth factors that stimulate cell proliferation and differentiation, and even metabolites, which are products of metabolism, utilize many of these exact same general mechanisms and signal transduction pathways. They are all interconnected, forming part of this vast, interwoven communication network that keeps us alive and functioning. And the clinical relevance here for anyone pursuing mastery in this field cannot be overstated. When there is an excessive, a deficient, or an inappropriate production of any of these signaling molecules, it becomes a major cause of disease. This is precisely why understanding these pathways at a deep level is so critical, because a vast number of modern pharmacotherapeutic agents, which are life-changing medicines, are aimed directly at influencing these very mechanisms to correct imbalances, restore function, and treat illnesses.
That's a perfect segue into understanding how these hormones and signaling molecules actually transduce their signals to affect homeostatic mechanisms. Let's walk through this process using a guiding framework, conceptually similar to Figure 42-1 from our source material. It starts, as everything in biology often does, with a stimulus. This isn't just a simple trigger, it's a challenge or a threat, whether to the entire organism, a specific organ, or even the fundamental integrity of a single cell. Think about it. At the organismic level, it could be something as dramatic as a sudden loud noise activating your nervous system, or a change in light recognized by your special senses. But equally important are the challenges at the cellular level. Physicochemical factors like a shift in pH, a change in oxygen tension, fluctuations in temperature, alterations in nutrient supply, the accumulation of noxious metabolites, or even changes in osmolarity, the concentration of solutes in the cell's environment.
So once that stimulus is present, the very first adaptive step the body takes is recognition. At the organismic level, as you highlighted, that's often the rapid response of the nervous system or the precise detection by our senses. But at the cellular level, specific receptors or highly specialized sensing mechanisms recognize these internal shifts or external cues. This appropriate recognition then leads to the precise hormone release. The body, with incredible specificity, releases one or more hormones or signaling molecules, each uniquely designed to orchestrate the necessary adaptive response to that specific stimulus.
After that precise release, we move into the crucial step of signal generation. This is where the magic, the molecular transformation truly happens. It's how the hormone's message is created, amplified, and conveyed at or within the target cells. And finally, you get the effects, the ultimate actions that take place inside the cell. These can be remarkably diverse, ranging from changes in gene transcription, which means altering which genes are turned on or off, to affecting transporters or ion channels on the cell membrane, leading to the physical translocation or movement of proteins to different parts of the cell, or even modifying existing proteins by adding or removing chemical tags. All of these effects then culminate in a finely coordinated response, the body's integrated and harmonized reaction to the initial stimulus. It truly is a beautifully orchestrated cascade, where each step leads seamlessly to the next, maintaining balance.
To understand this orchestration even more deeply, particularly how signals are generated and perceived, we categorize hormones. Based on where their specific cellular receptors are located and the type of signals they generate, our source, echoing Table 41-4, divides them into two main groups. First, we have group I hormones. These are characterized as lipophilic, meaning they are fat-loving. Because cell membranes are made of lipids, these hormones are often able to diffuse directly through the plasma membrane. They then interact with an intracellular receptor, meaning the receptor is located inside the cell, either in the cytoplasm or the nucleus.
And then we have group 2 hormones. These are often water-soluble, which means they can't just freely pass through the cell membrane like their group I counterparts. Instead, they interact with receptor recognition sites located on the extracellular surface of the plasma membrane. Think of it like a specific key fitting into a lock on the outside of a safe. This binding on the exterior then triggers a whole series of actions inside the cell. It's really worth reiterating here that molecules like cytokines, interleukins, and growth factors, while sometimes not traditionally listed as hormones in older textbooks, function very much like group 2 hormones. They are produced in specific cells, can have autocrine actions, meaning they act on the very cell that produced them, paracrine actions, acting on neighboring cells, or endocrine actions, traveling through the bloodstream to act on distant cells. Crucially, they bind to cell surface receptors and activate many of the same intricate signal transduction pathways as the more traditional group 2 hormones. So when we talk about group 2, understand that it encompasses a very broad and important family of signaling molecules. They are all part of the same complex signaling family, sharing fundamental mechanisms. So that's the grand overview. Now let's go inside the cell and unravel the mysteries of group I hormone action. This is where the initial binding event between the hormone and its receptor is the signal, directly initiating a cellular response. What's truly fascinating here, and a point of mastery for professionals, is how these lipophilic group I hormones can diffuse across all plasma membranes, freely entering virtually any cell they encounter. But here's where it gets really interesting. Despite being able to enter any cell, they only find their specific, high-affinity intracellular receptors in target cells. It's a remarkable level of precision.
This raises an important question for anyone trying to master this field. How does a cell know which hormone to respond to if these hormones just diffuse into every cell? The answer, as you precisely alluded to, lies entirely in the receptor. The receptor is like a highly specific molecular lock that only fits one particular key, the hormone. These receptors can be located either in the cytoplasm, the jelly-like substance filling the cell, or directly within the nucleus, the cell's command center. Once the hormone binds, this complex undergoes a crucial activation reaction. Let's take a detailed look at glucocorticoids as a prime example, following the left side of figure 42.2 from our source.
So a glucocorticoid, such as cortisol, diffuses right into the cytoplasm. There, it encounters its specific receptor, known as the glucocorticoid receptor, or GR. But here's an important twist. In its inactive state, the GR isn't just floating around freely. It's actually bound to a chaperone protein called heat shock protein 90, or HSP90. Think of HSP90 as a molecular placeholder or a protective cover, keeping the receptor at a particular inactive conformation and preventing it from prematurely interacting with DNA. It's like a sheathed sword, ready but not yet active.
Exactly. When the glucocorticoid hormone binds to the GR, it induces a significant conformational change in the receptor's three-dimensional shape. This change is absolutely critical because it causes the dissociation of that HSP90 chaperone, allowing it to unbind and float away. With HSP90 gone, a crucial segment on the GR, known as the nuclear localization sequence, or NLS, becomes exposed. This NLS acts like postal code or a molecular passport, facilitating the translocation, the movement of the now activated receptor ligand complex directly from the cytoplasm into the nucleus. Once securely inside the nucleus, this activated complex can then bind to specific DNA sequences to initiate its powerful effects on gene expression.
Now let's contrast that fascinating mechanism with another crucial example: thyroid hormones and retinoic acid, whose action is depicted on the right side of figure 42.2. They take a slightly different but equally elegant route. Instead of stopping in the cytoplasm and then translocating, they diffuse directly into the nucleus. And here's a crucial difference that marks a key distinction for professionals in this field. They're cognate receptors, meaning they're specific matching receptors, which are heterodimeric, meaning they're composed of two different protein subunits, often a thyroid receptor, TR, paired with a retinoid X receptor, RXR, are already bound to the hormone response element, or HRE, on the DNA, even in the absence of the hormone.
This is a truly elegant mechanism for regulating gene activity. In the absence of their specific ligand, these DNA-bound receptors exist in a complex with a corepressor. This corepressor acts as a tonic repressor of gene transcription, essentially keeping the gene off or silenced by modifying the chromatin structure. It's like a dampener on a piano string, preventing it from vibrating. However, when the ligand, be it thyroid hormone or retinoic acid, binds to its receptor, it induces another precise conformational change within the receptor. This change causes the dissociation of the corepressor or corepressors from the receptor, removing that inhibitory dampener. Once the corepressors are gone, the liganded receptor is now capable of binding coactivators with high affinity. This critical switch leads to the recruitment of other key players, including RNA polymerase II, the enzyme that synthesizes RNA, and general transcription factors, or GTFs. This entire assembly then culminates in the powerful activation of gene transcription, allowing the cell to produce new proteins. So instead of a direct on switch, it's a release from repression mechanism.
That brings us to a foundational concept for group I hormones, one that reveals the remarkable precision of gene regulation, the hormone response element, or HRE. These are specific DNA sequences that the hormone receptor complex recognizes and binds to. Table 42:1 in our source gives us some fantastic examples: the glucocorticoid response element, GRE, the thyroid hormone response element, TRE, the estrogen response element, ERE, among many others. You'll notice they often have these specific consensus sequences like GT-type and NPGT-TTT for the GRE. The N here means any nucleotide can be there, but the overall sequence, especially the repeating half sites, is incredibly specific. Often these are inverted palindromes where each half site binds one monomer of the receptor, like two hands clasping.
If we truly connect this to the bigger picture, especially for those aiming for mastery, the exact spacing between these half sites is absolutely key for generating specificity. Even with receptors that have very similar structures, this precise number of nucleotides separating the direct repeats dictates which hormone's receptor can bind effectively and trigger a response. For example, the vitamin D response elements, VDRE, thyroid hormone response elements, TRE, and retinoic acid response elements, RARE, all share a similar AGBTA half site. But the number of nucleotides separating those direct repeats, N3 for VDRE, N4 for TRE, and 5 for RARE, is what makes each element uniquely specific to its cognate receptor. This is an incredibly elegant and subtle mechanism for generating highly specific responses from what appear to be very similar DNA elements. And once the liganded DNA-bound receptor is in place, it acts as a high-affinity binding target for an army of transcriptional coactivator proteins, which then accelerate gene transcription, precisely turning on the right genes. We even see this principle at play with cyclic AMP's influence on gene transcription through its own binding element, the CRD.
It's incredible to think about the specificity and yet the scope of action here. Hormones typically affect less than 1% of the total genes, messenger RNAs, mRNA, or proteins in a target cell. Sometimes it's literally just a handful of genes that are specifically influenced. This truly highlights their surgical precision. While their dominant and most profound effect is on modulating gene transcription, hormones exhibit remarkable flexibility in how they exert their control. They can act at any step of what our source calls the information pathway, beautifully illustrated in figure 42-3. That pathway from gene to primary transcript, then to mRNA, and finally to protein, shows the flow of genetic information. Hormones can precisely influence transcription, the initial copying of the gene, translation, the synthesis of proteins from mRNA, modification, the chemical alteration of proteins, processing, their folding and maturation, transport their movement within or out of the cell, and even degradation, their controlled breakdown. This intricate control at multiple levels allows for fine-tuning specific gene expression and ultimately a precise biologic response.
And the understanding of hormone action doesn't start at the nucleus. Modern research has revealed direct actions of steroid hormones not just in the cytoplasm and on organelles like mitochondria, but even on cellular membranes, expanding their reach far beyond transcriptional control. Furthermore, modern discoveries continue to broaden our understanding of this intricate system. Recently, even microRNAs and lncRNAs, which are non-coding RNAs, meaning they don't directly code for proteins, have been implicated in mediating some of the diverse actions of hormones. This adds yet another layer of complexity and sophisticated control to this already intricate system, showcasing the continuous evolution of our mastery of this field.
So that's a deep dive into Group I hormones. Now let's pivot to Section 3. Membrane Mastery Group 2 Hormone Action in Intracellular Messengers. These Group 2 hormones have fundamentally different characteristics from Group I hormones. They are typically water-soluble, meaning they can't readily dissolve in the lipid bilayer of the cell membrane. As a result, they don't need transport proteins in the bloodstream, but they have a relatively short plasma half-life, meaning they are quickly broken down. And as we mentioned, they can't just diffuse through the plasma membrane to find an internal receptor. Instead, they must bind to receptors located on the outer surface of the cell's plasma membrane.
Precisely. Because they can't directly enter the cell, their mechanism of action relies on the generation of intracellular signals molecules often referred to as second messengers. Think of the hormone itself as the first messenger, the initial signal, and these molecules as the critical intermediaries that relay and massively amplify that signal inside the cell. Key second messengers we'll discuss include CAMP, cyclic AMP, cGMP, cyclic GMP, Ca2+, calcium ions, and a complex group called phosphatidylinosytides. The synthesis or release of these second messengers is triggered by the primary hormone binding to its receptor, and their impact is incredibly broad, affecting not only gene transcription but also a wide variety of other critical biological processes, ensuring the cell can respond rapidly and comprehensively.
Let's begin with a major player in this field, one that revolutionized our understanding of cell signaling. G-protein coupled receptors, or GPCRs, and their crucial connection to CAMP. GPCRs are the single largest family of cell surface receptors in humans. Structurally, they are described as typically having 7 ahelical hydrophobic domains that span the plasma membrane, meaning they weave back and forth through the cell membrane 7 times. You can visualize them as those 7 interconnected cylinders extending through the lipid bilayer in figure 42-4 of our source.
Here's where it truly gets fascinating for anyone seeking mastery: the sheer significance of GPCRs. Hundreds of GPCR genes have been identified, making them an overwhelmingly vast family of cell surface receptors. This astonishing diversity means they mediate an absolutely huge variety of physiological responses throughout the human body, everything from your sense of vision and taste to regulating mood, controlling immune function, and orchestrating crucial metabolic processes. And because of their pervasive and fundamental roles in so many bodily functions, they are, without question, major drug targets for countless pharmacotherapeutic agents. Understanding their intricate dance is absolutely key to understanding modern medicine and drug development.
And these GPCRs, despite their inherent complexity, don't act alone. They operate through a crucial intermediary, the G protein. In its inactive state, before a hormone is bound to the receptor, the G protein is a heterotrimeric complex, meaning it's made of 3 distinct protein subunits, alpha, beta, and gamma. In this resting state, it's bound to a molecule called GDP, and it's likely not directly associated with the receptor. It's anchored to the plasma membrane by specific lipid groups, patiently waiting.
But when a hormone binds to the receptor, that's when the dynamic activation occurs. First, the hormone H binds to its specific receptor, inducing significant conformational changes in the receptor structure. This rearranged receptor then physically associates with the G protein complex, effectively activating it. The most critical step here is the exchange of GDP for GTP on the HS subunit. This switch from TDP to GTP energizes the HS subunit. Once the HS subunit is bound to GTP, it physically dissociates from the H complex, becoming an active signaling entity. This active IGTP subunit then travels to and binds to an effector E, which could be an enzyme like adenylyl cyclase or an ion channel or another protein as illustrated in figure 42.4 and detailed in table 42.3. It's also crucial to remember that the fake subunit isn't just a passive bystander after dissociation. It too can have direct actions on various effectors, adding another layer of signaling complexity.
Now let's zoom in on one of the most well-known effectors, adenylyl cyclase, and its role in cGMP production. Cyclic AMP, or cAMP, holds a special place in history as the very first intracellular second messenger signal identified in mammalian cells. Different peptide hormones can either stimulate or inhibit the production of cAMP from adenylyl cyclase. We have what are called stimulatory systems, often denoted by Gs, where E stands for stimulatory, which are activated by hormones like ACTH, ADH, and glucagon. And then there are inhibitory systems, denoted by G, where Ni stands for inhibitory, which are activated by molecules like acetylcholine and adrenergics, as shown in Table 42. Despite their opposing effects, they all ultimately converge on the adenylyl cyclase enzyme to regulate cAMP levels.
And the G protein subunits themselves demonstrate astonishing diversity, a complexity that allows for incredibly fine-tuned control. As Table 42-3 indicates, there are at least 21 identified S subunit genes, 5 S subunit genes, and 8 S subunit genes. This leads to a vast number of possible combinations, each with potentially distinct functions and specificities, allowing for an immense range of cellular responses. The SS protein, which is part of the stimulatory G protein, possesses intrinsic GT-PACE activity. This is a built-in molecular off-switch absolutely essential for ensuring rapid and precise responses. The active form, SSGTP, is swiftly inactivated when its bound GTP is hydrolyzed back to GDP. This chemical change causes the trimeric Gs complex to reform, ready for the next signaling cycle.
This built-in regulation is so precise, and remarkably, we actually gained immense insights into its mechanisms by studying toxins. For instance, cholera toxin, famously responsible for severe diarrheal disease, ADP ribosylates the active subunit. This chemical modification completely disrupts its intrinsic GT-PACE activity. Without this off switch, ACEs can't reassociate with PEG and remains irreversibly activated, leading to persistent, uncontrolled cAMP production. Think of the severe watery diarrhea caused by cholera that's partly due to this relentless cAMP signaling in intestinal cells, causing excessive fluid secretion. Similarly, pertussis toxin, responsible for whooping cough, ADP ribosylates out too, preventing its dissociation from PONS, thus leaving ACEs activity unopposed. These toxins, by disrupting normal function, teach us so much about the intricate built-in control mechanisms of these pathways. For a professional, understanding these perturbations is key to appreciating the normal ballet.
The functions of G proteins are incredibly diverse, extending far beyond simply regulating adenylyl cyclase. As Table 42-3 clearly shows, their roles are pervasive. For instance, some isoforms can stimulate K+ channels, leading to hyperpolarization, and inhibit Ca2+ channels, reducing calcium influx. Conversely, some ICE molecules have the opposite effects on Ca2+ channels. Members of the GQ family activate the phospholipase C group of enzymes, which we'll discuss in detail in a moment, leading to different second messenger pathways. And even the IR complexes themselves, once dissociated from the IS subunit, can directly stimulate K+ channels and activate phospholipase C, adding another layer to the signaling network. Beyond hormonal signaling, G proteins are crucial in sensory processes like olfaction, mediated by ILF, and vision, mediated by IS. Their widespread involvement across so many physiological processes means GPCRs are implicated in numerous diseases and are, consequently, primary targets for a vast number of pharmaceutical agents.
So we have cAMP being produced. But what does it do inside the cell? That's where protein kinase A, or pKa, comes in, serving as the crucial cAMP effector, as beautifully illustrated in Figure 42-5. pKa exists as an inactive heterotetrameric molecule, meaning it's composed of 4 parts, 2 regulatory R subunits and 2 catalytic C subunits, bound together as R2C2. In this bound state, the catalytic subunits, which are the enzymes responsible for phosphorylation, are inhibited, essentially held in check.
Here's a true aha moment, a fundamental concept for professional understanding. When cAMP is produced in response to a hormonal signal, 4 molecules of cAMP specifically bind to the 2 regulatory subunits of pKa. This binding induces a critical conformational change in the regulatory subunits, which then causes the dissociation of the RC complex, thereby activating the catalytic C subunits. These now active C subunits spring into action, catalyzing the transfer of the terminal gamma phosphate from ATP to specific serine or threonine residues within a variety of target proteins. They recognize very precise consensus phosphorylation sites, often sequences like Argolese X Surther, ensuring that only the correct proteins are modified.
And this phosphorylation isn't just a specific event for pKa. Protein phosphorylation, the addition of a phosphate group, is now recognized as a major, widespread, and ubiquitous regulatory mechanism throughout virtually every cell in your body. Hundreds of different protein kinases exist, each with unique specificities for which proteins they phosphorylate and on which amino acid residues. What's really clever and crucial for precise cellular responses is how these kinases are localized within the cell. Akinase anchoring proteins, or AKPs, act as molecular scaffolds or docking stations that precisely localize pKa near its specific substrates. This ensures that pKa activities focus on the right physiological targets at the right time and place, allowing common signaling proteins to elicit highly specific responses. AKCalers are truly multifunctional, binding pKa, but also other kinases, phosphatases, and even phosphodiesterases, which enhances the precise localization, the rate, the specificity, and the dynamic control of the signaling pathway.
For those aiming for mastery, it's critical to understand that all of the diverse effects of cGMP in eukaryotic cells are thought to be mediated by this precise process of protein phosphorylation and dephosphorylation, primarily on serine and threonine residues. This means that the ultimate specificity of a cellular response, whether it's dehydrogenesis, ion transport, carbohydrate or fat metabolism, enzyme induction, gene regulation, or synaptic transmission, is conferred not just by the initial hormone signal, but by the specific protein kinase activated, the specific phosphatase that reverses its action, and most importantly, the specific target substrates that are being phosphorylated in that particular cell type. The precise array of these specific substrates is what truly defines a target tissue and dictates the magnitude and nature of a particular response within a given cell.
A prime real-world example of CAMP's profound impact on gene transcription is through the cyclic AMP response element binding protein, or CRE. When TREAT binds to a specific DNA enhancer element known as a CRE, or cyclic AMP response element, in its non-phosphorylated state, it's actually a pretty weak activator of gene transcription. It's like an engine running in neutral. But when pKa phosphorylates CR at key amino acid residues, it triggers a powerful change. This phosphorylation causes CR to bind to a potent coactivator called CR binding protein, or CBP, and its close relative P300. As a result, this complex becomes a much more potent transcriptional activator, essentially putting the engine in gear. And here's another crucial detail for deep understanding. CBP and P300 contain intrinsic histone acetyltransferase activities, known as HATs. This means they are chromatin-active transcriptional coregulators, directly helping to remodel chromatin, the tightly packed DNA and protein structure within the nucleus, to make genes more accessible for transcription. They can even acetylate certain transcription factors, further stimulating their ability to bind DNA and modulate gene expression.
Just as important as turning signals on is having the sophisticated mechanisms to turn them off, and that's where signal termination comes in. Actions caused by hormones that increase SAMP concentration are terminated in a couple of key, elegant ways. First, enzymes called phosphodiesterases hydrolyze SAMP, breaking it down into an inactive molecule called 5-AMP. This action ensures a rapid turnover of the signal, SAMP, and therefore a rapid termination of the biological process once the hormonal stimulus is removed. This is absolutely crucial for the body to maintain precise control, not just having powerful on switch, but also a swift and efficient off switch. There are at least 11 members in this phosphodiesterase family, each with distinct regulation influenced by their substrates, SAMP and cGMP, by hormones themselves, and by intracellular messengers like calcium. Interestingly, inhibitors of phosphodiesterase, such as methylated xanthine derivatives like caffeine, increase intracellular CAMP levels by preventing its breakdown, effectively mimicking or prolonging the actions of hormones through this signaling pathway. This explains some of caffeine's stimulant effects.
The second critical control mechanism involves phosphoprotein phosphatases. These enzymes are the counterparts to kinases. They regulate the dephosphorylation reaction, precisely reversing the work of the kinases by removing phosphate groups from proteins. Like kinases, phosphatases themselves are subject to incredibly fine-tuned regulation through phosphorylation-dephosphorylation reactions and intricate protein-protein interactions. Their substrate specificity, meaning which proteins they act on, is often dictated by distinct regulatory subunits, which can themselves be hormonally regulated. A classic, well-studied example is in glycogen metabolism in muscle, where different types of phosphatases like type 1 and type 2, along with their heat-stable inhibitors, precisely control the dephosphorylation of key enzymes involved in glycogen synthesis and breakdown. And for an even broader picture, phosphatases that specifically target phosphotyrosine residues are also incredibly important in signal transduction, playing crucial roles we'll discuss shortly.
Moving on to another crucial intracellular signal, one that operates similarly to CAMP but with distinct functions, cGMP. This molecule, cyclic GMP, is made from GTP by the enzyme guanylyl cyclase, which exists in two distinct forms: a soluble form found in the cytoplasm and a membrane-bound form, each with unique physiological properties. The membrane-bound guanylyl cyclase is specifically activated by atriopeptins, a family of cardiac peptides like atrial natriuretic factor, which are produced in the heart. These peptides cause natriuresis, which is the excretion of sodium by the kidneys, diuresis, which is water excretion, vasodilation, the widening of blood vessels, and inhibition of aldosterone secretion, a hormone that retains sodium. So a peptide from your heart can directly influence your kidney function and blood pressure by increasing cGMP by as much as 50-fold.
And then there's the soluble guanylyl cyclase, which is primarily activated by gaseous compounds like nitric oxide and clinically relevant agents like nitroprusside and nitroglycerin. These agents are powerful vasodilators and cause smooth muscle relaxation throughout the body. This is a great example, for those interested in therapeutic applications, of how understanding these fundamental pathways leads directly to the development of new medicines. For instance, inhibitors of cGMP phosphodiesterase, such as the drug sildenafil, widely known as Viagra, work precisely by preventing the breakdown of cGMP, thereby enhancing and prolonging these vasodilatory responses. The increased cGMP then activates cGMP-dependent protein kinase, or PKG, which in turn phosphorylates specific proteins in smooth muscle, leading to relaxation and vasodilation.
Next, let's turn our attention to calcium, Ca2+, which is truly a universal regulator throughout biology. It's involved in an astonishingly massive range of cellular and physiological processes, from muscle contraction and stimulus secretion coupling like the release of neurotransmitters or hormones, to critical functions like blood clotting, modulating enzyme activity, and regulating membrane excitability. Critically, Tal-2+ also serves as a crucial intracellular messenger for hormone action, relaying and amplifying signals from the cell surface to internal machinery.
To achieve this universal regulatory role without causing cellular damage, the body maintains incredibly tight and precise control over calcium metabolism. Extracellular Ca2+ concentration is maintained around 5 millimolar, but intracellular free Ca2+ is kept extraordinarily low, typically between 0.05 and 10 micromolar. That's a truly massive 10,000-fold or more concentration gradient. Despite this steep gradient and a favorable electrical gradient across the cell membrane that would normally drive calcium into the cell, Ca2+ is actively restrained from entering. A considerable amount of cellular energy is expended just to control intracellular Ca2+ levels, because a prolonged elevation of Ca2+ inside the cell is actually very toxic and can trigger cell death.
To manage this critical balance, cells have evolved several sophisticated mechanisms for rapidly removing Ca2+ from the cytosol. There's a high-capacity but low-affinity Na plus Ca2+ exchange mechanism that effectively extrudes Ca2+ from the cell, pushing it out into the extracellular space. Then there's a Ca2+ proton ATPase-dependent pump, which has a very high affinity for Ca2+ but a lower capacity, likely responsible for the fine-tuning of cytosolic Ca2+ levels. Furthermore, specific Ca2+ ATPases actively pump Ca2+ from the cytosol into the lumen of the endoplasmic reticulum, a network of membranes within the cell where it's stored in large intracellular pools. Cytosolic Ca2+ levels can be rapidly changed in three main ways in response to hormones. 1. Some hormones, by binding directly to specific receptors that are Ca2+ channels, directly enhance membrane permeability, rapidly increasing Ca2+ influx. 2. Hormones can also indirectly promote Ca2+ influx by modulating the cell's membrane potential, which then opens voltage-gated Ca2+ channels. 3. And, crucially, K2+ can be mobilized rapidly from those vast intracellular stores, primarily from the endoplasmic reticulum and possibly from mitochondrial pools as well.
Now, once calcium levels change, how does the cell sense this and translate it into a response? That's where calmodulin comes in, acting as the cell's sophisticated calcium sensor. The discovery of calmodulin, a Ca2+ attendant regulator of phosphodiesterase activity, was a huge breakthrough in understanding how Ca2+ and CAMP signaling pathways often interact. Calmodulin is a relatively small 17 kilodalton protein, structurally and functionally highly homologous to the muscle protein troponin C. It has 4 specific Ca2+ soluble binding sites. When all 4 sites are occupied by calcium, it undergoes a marked conformational change, like a molecular hand tightening its grip, which then allows it to activate a remarkable variety of enzymes and ion channels. This concept, Ca2+ binding to calmodulin and changing its activity, is conceptually very similar to how CAMP binding activates pKa, showcasing a common regulatory theme in biology.
Calmodulin is not just a passive sensor, it's an active regulatory protein, often serving as a crucial subunit of larger protein complexes. It's particularly involved in regulating various kinases and enzymes related to cyclic nucleotide generation and degradation, linking calcium signals to other pathways. Table 42.4 lists some of the many enzymes and proteins regulated directly or indirectly by Ca2+A, most likely through calmodulin, including adenyl cyclase, various Ca2+ dependent protein kinases, phosphodiesterase, and even some critical ion channels and nitric oxide synthase. Beyond enzymes, Kina2 plus calmodulin also regulates vital structural elements, such as the actin-myosin complex in smooth muscle, which drives contraction, and microfilament-mediated processes in non-contractile cells, like cell motility, changes in cell shape, the processes of mitosis, and the release of cellular granules.
There's overwhelming evidence that calcium is a crucial and often universal mediator of hormone action. Many hormonal effects are significantly blunted by experimental conditions involving CO2 plus zeta-free media or when intracellular calcium is depleted. Conversely, these very effects can be mimicked by agents that increase cytosolic CO2 plus 8, such as the Ca2 plus ionophore A23187, a chemical that literally carries calcium across membranes. Hormones also directly influence cellular calcium flux, either by opening channels or releasing stored calcium. A classic, well-studied example is the regulation of glycogen metabolism in the liver by vasopressin and butyrate catecholamines, where calcium plays a central role. A number of critical metabolic enzymes, including glycogen synthase and pyruvate kinase, are regulated by Ca2 plus Ca, by phosphorylation, or often by both, highlighting the intricate crosstalk within the cell.
The question then becomes, for those seeking true mastery, how does a signal from a membrane receptor effectively communicate with these internal Ca2 plus reservoirs within the cell, particularly the endoplasmic reticulum? The elegant solution lies in the products of phosphatidylinoxidase metabolism, which act as critical intermediaries and powerful second messengers, as beautifully illustrated in Figure 42-6 and 42-7. Cell surface receptors, such as those for acetylcholine, antidiuretic hormone, ADH, and I1-type catecholamines, when occupied by their specific ligands, become potent activators of an enzyme called phospholipase C, or PLC. This receptor binding and subsequent PLC activation are precisely coupled by the GQ isoforms of G proteins, as we discussed earlier when detailing the diverse functions of G protein subunits in Table 42.3.
So when PLC is activated, it performs a crucial enzymatic step. It catalyzes the hydrolysis or breakdown of a specific membrane lipid called phosphatidylinoxidase, 4-involved bisphosphate or PIP2, into 2 distinct and incredibly crucial second messengers, inositol trisphosphate, IP3, and 1-methyl-2-diacylglycerol, DAG. This is where the molecular connection truly forms. IP3, being water-soluble, diffuses into the cytoplasm and interacts with a specific intracellular receptor, primarily located on the endoplasmic reticulum. This interaction causes the rapid release of Ca2+ from these internal storage sites, dramatically increasing cytoplasmic calcium levels. Meanwhile, DK, the other product, remains embedded in the membrane and directly activates protein kinase C, or PKC, an enzyme whose activity also crucially depends on the presence of calcium.
So you have this remarkable convergence. The hydrolysis of PIP2 simultaneously leads to the direct activation of PKC and a significant increase in cytoplasmic Ca2 plus CO. And as we saw back in Figure 42.4, G proteins can also directly influence Ca2 plus channels, further impacting intracellular Ca2 plus levels. The resulting elevations of cytosolic Ca2+ activate not only Ca2+ calmodulin-dependent kinases, but a whole host of other Ca2+ calmodulin-dependent enzymes. Simultaneously, the activated PKC phosphorylates its own unique set of protein substrates. Both of these powerful pathways, initiated by the same membrane event, ultimately lead to profound and altered physiological processes. We see examples of this complex interplay across many systems. ACTH and CAMP in the adrenal cortex, angiotensin II and other factors in the zona glomerulosa, and LH and CAMP in the ovary and Leydig cells, all of which are associated with increased levels of phosphatidic acid and phosphonosides in their target tissues, signifying this intricate interconnected dance of molecules.
Now let's move into Section 4. Kinase cascades amplifying and diversifying signals. We've discussed individual protein kinases like pKa, pKc, and various KMKs, which are powerful enzymes that add phosphate groups to serine and threonine residues on proteins. They're incredibly important for basic cellular regulation. But what truly allows for massive signal amplification, incredible diversification of signals, and highly integrated cellular responses is the concept of kinase cascades, where multiple protein kinases act sequentially, one activating the next, much like a series of molecular dominoes.
A major conceptual breakthrough in this area was the discovery that the epidermal growth factor, or EGF receptor, possesses intrinsic tyrosine kinase activity. That means the receptor itself, when bound by its ligand, doesn't just pass a signal. It is an enzyme capable of phosphorylating other proteins, specifically on their tyrosine residues. The insulin receptor and insulin-like growth factor 1, IGF-1 receptors, also contain this intrinsic ligand-activated tyrosine kinase activity. What distinguishes these pathways, and is critical for professional understanding, is threefold. First, these kinases preferentially phosphorylate tyrosine residues, which is much less common than serine phosphorylation in mammalian cells, making it a very specific and impactful signal. Second, tyrosine phosphorylation is relatively infrequent overall, accounting for less than 0.03% of total amino acid phosphorylation, underscoring its precise signaling importance. Third, and most importantly, the ligand-receptor interaction doesn't just trigger a single phosphorylation event. It initiates a complex cascade involving multiple protein kinases, phosphatases, and other regulatory proteins, leading to widespread, amplified, and coordinated effects throughout the cell.
Let's really dig into this with a prime example. Insulin, which transmits signals by several sophisticated kinase cascades, beautifully summarized in Figure 42.8. The initial activation begins when insulin binds to its specific receptor, the insulin receptor, or IR. The IR is quite a complex molecule itself, a heterotramer, meaning it's made of 4 distinct protein subunits. This banding event activates the IR's intrinsic tyrosine kinase activity, leading to a crucial step called autophosphorylation. This means the IR phosphorylates itself on specific tyrosine residues.
These newly phosphorylated tyrosines on the activated insulin receptor then serve as crucial docking sites, like molecular plugs fitting into specific sockets. The activated receptor then goes on to phosphorylate a family of adapter molecules called insulin receptor substrates, or IRS1 to 4, also on their tyrosine residues. These phosphorylated IRS molecules, now activated themselves, then bind to specialized protein domains called cellular C homology 2, or SH2 domains. These SH2 domains are like molecular hooks or specialized clamps that specifically grab onto these activated phosphorylated tyrosine residues. This crucial binding then mediates insulin's incredibly diverse effects by linking the IRS proteins to various downstream signaling molecules.
One of the most important and well-studied branches originating from this point is pathway 1, the PI3 kinase pathway, which is absolutely crucial for many of insulin's metabolic actions. When PI3 kinase specifically binds to a phosphorylated IRS molecule via its SH2 domain, it becomes activated. PI3 kinase then, remarkably, generates novel inositol lipids. These are unique lipid molecules that act as their own distinct second messengers within the cell. These newly generated lipids, in turn, activate another crucial enzyme, phosphoinositide-dependent kinase 1, or PDK1. PDK1 then acts as a central hub, propagating the signal to other key kinases downstream, including protein kinase B, also widely known as AKT, SGK, and a group of atypical PKCs. There's also a distinct alternative branch involving P70S6K and other kinases, showing the pathway's branching complexity.
A key regulator in that alternative P70S6K branch is mTOR, or mammalian target of rapamycin. mTOR is a major nutrient sensor directly regulated by amino acid levels and significantly by insulin. It's essential for P70S6K activity. This mTOR signaling system helps distinguish between the PKB-AKT and P70S6K branches downstream from PDK1, adding another layer of regulatory finesse. The overall outcomes of this pathway are incredibly important for insulin's functions and metabolism. This includes the rapid protein translocation of glucose transporters, like GLUT4, to the cell surface, allowing cells to take up glucose, as well as the activation of key enzymes and the precise regulation of genes involved in metabolism, such as peptidase, hexokinase 2, and glucokinase. It's a master switch for how cells handle energy.
The second major pathway activated by insulin is the MAK pathway, which stands for a mitogen-activated protein kinase. This pathway is primarily involved in crucial cellular processes like proliferation, meaning cell growth and division, and differentiation, where cells specialize into different types. In this pathway, another SH2 domain-containing protein called GRB2 binds directly to IRS1. This binding event serves as a crucial molecular link, connecting the initial tyrosine phosphorylation signal to the activation of a cascade of threonine and serine kinases. Specifically, it leads to the activation of a small GT-PACE protein called P21-rase. Activated P21-rase then initiates a sequential protein kinase cascade. It activates RAF1, which then activates MEK, which in turn activates the P42, P44 MEP kinase isoforms, also known as ER. The ultimate outcomes of this pathway are critical for the precise regulation of proliferation and differentiation in many cell types, driving growth and development.
It's absolutely essential to remember that all these phosphorylation events, as powerful as they are reversible. And therefore, signal termination is critical for precise control. Specific phosphatases, like PTAN for the product of the PI3 kinase reaction, actively dephosphorylate these molecules, thereby antagonizing the pathway and terminating the signal. This acts as an indispensable off switch to ensure that responses are transient and tightly regulated. And interestingly, insulin also indirectly affects many enzymes by activating specific phosphodiesterases, which in turn reduces intracellular cGMP levels, adding another subtle but significant layer to its already complex signaling network, allowing it to fine-tune many metabolic processes.
Another vital kinase cascade, distinct yet equally important, involves the JAK-STAT pathway, used by many cytokines and growth factors, as shown in Figure 42-9. What's unique here, and a point of distinction for professionals, is that the receptors for hormones like growth hormone, prolactin, erythropoietin, and many crucial cytokines lack intrinsic kinase activity themselves. They're not like the insulin receptor with its built-in tyrosine kinase domain. So how do they signal?
They employ a clever workaround. When the ligand, the signaling molecule, binds to its receptor, it causes the receptor to dimerize, meaning two receptor units come together. This dimerization then brings into close proximity and activates associated cytoplasmic protein tyrosine kinases. These are a specific family of kinases known as Janus kinases, or JAKs, such as JAK1, JAK2, or tyrosine kinase 2, TYK2. Once activated, these JAKs, in turn, phosphorylate the receptor itself on specific tyrosine residues, creating new docking sites.
These newly phosphorylated tyrosine residues on the receptor then become perfect docking sites for another crucial family of cytosolic proteins called STATs, which stands for signal transducers and activators of transcription. The STATs specifically associate with the phosphorylated receptor via their own SH2 domains, those molecular hooks we discussed earlier. Once docked, the STATs are then themselves phosphorylated by the active JAKs. Upon phosphorylation, the STAT proteins dimerize, meaning two STAT molecules come together, and then this active dimer rapidly translocates into the nucleus. Once inside the nucleus, the STAT dimer binds to specific DNA elements, such as the interferon response element, IRE, and directly activates gene transcription, turning on genes critical for processes like immune responses and cell growth.
What's truly fascinating here, and a hallmark of sophisticated biological systems, is the potential for crosstalk. The phosphorylated tyrosine residues on the receptor aren't exclusive to STATs. They can also bind other SH2 domain-containing proteins. These other docking events can activate entirely different pathways, such as the PI3 kinase pathway or the MAP kinase pathway via adapter proteins like SHC or GRB2. They can even lead to G protein-mediated activation of phospholipase C, PLC, with subsequent DAG production and PKC activation. This intricate web of interactions demonstrates how different hormones or signaling molecules can talk to the same fundamental cellular machinery, leading to incredibly complex, integrated, and finely tuned responses, where multiple signals converge or influence each other.
Let's look at another incredibly critical pathway, especially in the context of inflammation, the NFU pathway, and how it's elegantly regulated by glucocorticoids as depicted in Figure 4210. NFU is a heterodimeric DNA-binding transcription factor, typically composed of two protein subunits, P50 and P65. Its primary vital role is to regulate the transcription of genes that are crucial for initiating and orchestrating the inflammatory response, making it a central player in immunity and inflammation.
Normally, in a resting cell, NFQ is kept in an inactive state, carefully sequestered in the cytoplasm. This is achieved through its interaction with a family of proteins called IB, or inhibitor of NFQ. IB essentially acts like a molecular straitjacket, masking NFQ's nuclear localization signal. This prevents NFQ from entering the nucleus and activating gene transcription, keeping inflammation in check under normal conditions.
However, various powerful extracellular stimuli, such as pro-inflammatory cytokines like TNFi, reactive oxygen species, and mitogens, can trigger the activation of the IKK complex, which is a large heterohexameric structure containing multiple subunits. This activated IKK then performs a critical phosphorylation step. It phosphorylates IgA on two specific serine residues. Once IE is phosphorylated in this manner, it's immediately targeted for polyubiquitylation, a process where multiple ubiquitin tags are attached to it. This ubiquitin tag acts like a molecular flag, marking IE for subsequent degradation by the proteasome, which is essentially the cell's sophisticated protein recycling plant. With IE rapidly degraded and out of the way, the previously masked NFA is now free and rapidly translocates into the nucleus. There, it binds to various gene enhancers and robustly activates transcription, particularly of those powerful inflammatory genes. This transcriptional regulation is often aided by coactivators like the Sierra binding protein, or CBP, which we discussed earlier.
This brings us to the profound clinical significance of glucocorticoids, which are incredibly potent anti-inflammatory and immunomodulatory agents, widely used therapeutically for conditions ranging from asthma to autoimmune diseases. Their remarkable ability to quell inflammation is explained, in large part, by their intricate and multi-pronged inhibition of NFB. For those striving for a mastery-level understanding, evidence points to three main, elegant mechanisms for this inhibition. First, glucocorticoids actually increase the expression of IDMRNA, which leads to more IB protein being produced. This results in more efficient sequestration of NFB in the cytoplasm, preventing its nuclear entry. Second, the glucocorticoid receptor, or GR, directly competes with NFP for binding to essential coactivators, essentially limiting NFP's ability to activate genes. And third, the GR can directly bind to the P65 subunit of NFE itself and inhibit its activation. This is a beautiful and sophisticated example of how one powerful signaling pathway can directly interfere with another to achieve a potent and widespread therapeutic effect, showcasing the body's natural checks and balances and how we leverage them in medicine.
We've now explored these incredible molecular dances, these intricate cascades and signaling pathways. But what's the ultimate goal? What is the final destination for so many of these signals? It's all about the nuclear command center modulating transcription. Ultimately, all these diverse signals, generated from distinct pathways, converge and lead to precise changes in gene transcription rates within the nucleus, enabling the cell to effectively adapt to physiological challenges and maintain homeostasis.
Many fundamental observations have guided our deep understanding of how hormones ultimately affect transcription. For one, we know that actively transcribed genes are found in regions of open chromatin, the highly organized structure of DNA and proteins within the nucleus. This openness is often indicated by sensitivity to enzymes like DNase-cal and by specific histone post-translational modifications, PTMs, which make the DNA accessible for gene expression. We also understand that genes have specific regulatory regions where transcription factors bind to precisely modulate the frequency of transcription, whether it's at the initiation of the RNA synthesis, promoter clearance, or the elongation phase. The hormone receptor complex itself can be one of these crucial transcription factors, binding directly to specific hormone response elements, or HREs, on the DNA. Alternatively, other signals generated by hormones through those intricate cascades we just discussed can modify the location, the amount, or the activity of existing transcription factors, thereby influencing their binding to regulatory elements. Furthermore, there's a large superfamily of nuclear receptors that act with, or analogously to, the hormone receptors we've discussed, sharing similar mechanisms of action. And critically, these nuclear receptors interact with another large and diverse group of co-regulatory molecules to effect precise changes in the transcription of specific genes.
Let's revisit HREs, because several have been precisely defined, providing a blueprint for gene regulation. They exhibit properties similar to enhancer elements in that their function isn't strictly dependent on their exact position, their specific location relative to the gene, or even their orientation on the DNA. While generally found upstream of the transcription initiation site, they can also be located within the coding region of the gene, or even in introns, the non-coding sequences within a gene. The identification of these HREs was achieved using clever experimental strategies, like heterologous reporter systems where the HRE is linked to an easily measurable gene, and modern high-throughput techniques like genome-wide chromatin immunoprecipitation sequencing, ChIP-seq, and sophisticated bioinformatic analyses, as outlined in chapters 38 and 39 of our source. These methods allow scientists to pinpoint exactly where transcription factors bind across the entire genome.
But here's where it gets truly complex, and was a major puzzle for scientists in the early days, driving much of our current understanding. Glucocorticoids, progestins, mineralocorticoids, androgens, remarkably, they all use the same HRE consensus sequence as clearly shown in Table 42:1. So how do they achieve their vastly different physiological actions when their DNA binding sites are identical? This was a profound biological riddle. It became clear that the regulatory circuitry of natural genes is far more intricate than simply a hormone receptor binding to a single HRE. The HREs are almost always found associated with other specific regulatory DNA elements and their associated binding proteins, and these additional associations are absolutely mandatory for optimal function and specificity. The extensive sequence similarity, particularly in the DNA binding domains between the classic steroid hormone receptors, ultimately led to the groundbreaking discovery of the nuclear receptor superfamily of proteins. These receptors, along with a large number of accompanying co-regulator proteins, allow for a wide variety of DNA protein and protein interactions, providing the exquisite specificity necessary for highly regulated physiological control. Figure 4211 provides a fantastic schematic of such a complex assembly, which is called the Hormone Response Transcriptional Activation Unit.
This hormone response transcriptional activation unit is quite a marvel of molecular engineering. It involves the core HRE, which is bound by the ligand-bound receptor R. But crucially, there are also adjacent accessory factor elements, or AFEs, with their own specific bound transcription factors. More than 24 of these accessory factors, which are often themselves members of the nuclear receptor superfamily, have been linked to hormone effects on transcription. These AFs can interact with each other, with the liganded nuclear receptors, or with various co-regulators.
And all these components then communicate seamlessly with the basal transcription machinery, including RNA polymerase II and the general transcription factors, through a large, sophisticated complex of co-regulators. This co-regulator complex can include members from various families, such as the P160 family, corpressor family, mediator-related proteins like TRAPS and DRIPS, or the CBPP300 families, as detailed in Table 42-6. Many of these transcription co-regulators possess intrinsic enzymatic activities that are absolutely essential for gene regulation. They can covalently modify the DNA itself, various transcription proteins, and critically, the histones present in the nucleosomes, which are the protein spools around which DNA is wound. This enzymatic activity essentially remodels the chromatin, making the DNA more accessible for transcription. Collectively, the hormone, its specific receptor, the dynamic chromatin structure, the underlying DNA sequence, and the intricate transcription machinery all integrate and process hormone signals in a highly coordinated fashion to precisely regulate transcription, achieving perfect physiological harmony. It's truly an orchestra playing in a perfect harmony.
This brings us to an incredibly important concept for anyone seeking true mastery in this field. There is a large family of nuclear receptor proteins. This superfamily consists of a diverse set of transcription factors, initially discovered due to compelling sequence similarity in their DNA binding domains. This family now boasts more than 50 known members, including not only the classic nuclear hormone receptors we've been discussing, but also many fascinating orphan receptors, for which the natural physiological ligand has yet to be discovered. They're like receptors still waiting for their key.
These nuclear receptors, despite their diverse functions, share several fundamental common structural features, which allow us to categorize and understand them, as shown in Figure 4212. They all possess a centrally located C domain, which is the absolutely critical DNA binding domain, DBD. This domain contains two unique zinc finger binding motifs that directly guide the receptor's specific binding to DNA. They can bind to DNA as homodimers, two identical receptor units, as heterodimers, typically with a retinoid X receptor or RXR as a partner, which acts as a versatile common partner for many, or even as monomers, depending on the specific receptor. The target response element on the DNA consists of one or two DNA half-site consensus sequences precisely arranged as an inverted or direct repeat. What's truly crucial and an incredibly elegant mechanism for fine-tuning specificity is that the spacing between the direct repeats determines the binding specificity. For example, a direct repeat with 3 nucleotide spacers, N3, specifies the binding of the vitamin D receptor, VDR. N44 specifies the thyroid hormone receptor, TR. And NA5 specifies the retinoic acid receptor, RAR, all potentially binding to the same fundamental consensus DNA half-site, Table 42-1. This spacing is a molecular trick for generating highly specific responses from similar DNA elements, a testament to evolutionary efficiency.
Then there's the E domain, which is the ligand binding domain, LBD, located at the carboxyl terminus, the end of the protein. This domain is remarkably multifunctional. It selectively binds specific hormones and metabolites, and this binding is what dictates the particular biological response. The LBD also contains crucial subdomains for binding heat shock proteins, mediating the dimerization of the receptor subunits, enabling its nuclear localization and facilitating its transactivation. This latter function is specifically facilitated by a carboxyl terminal transcription activation function, known as the AF2 domain, which forms a surface crucial for interaction with coactivators.
A highly variable D region, or hinge region, separates the DBD from the LBD, providing essential flexibility to the receptor, allowing it to adapt its shape and interactions. And finally, there's a highly variable AB domain, located at the amino terminal region. This domain contains another activation domain called AF1. The AF1 domain is thought to contribute to distinct physiological functions by binding different co-regulator proteins. This region of the receptor can also give rise to different receptor isoforms through the use of different promoters that initiate transcription, alternative splice sites that combine different RNA segments, or even multiple translation initiation sites. These isoforms share identity in their DBD and LBD, but because they associate with various coregulators through this variable amino terminal AF1 domain, they can exert very different physiological responses, adding yet another layer of regulatory complexity.
We can classify this large family of receptors based on how they bind to their respective DNA elements, as comprehensively outlined in Figure 4212 and Table 42-5. The classic steroid hormone receptors like the glucocorticoid receptor, GR, mineralocorticoid receptor, estrogen receptor, ER, androgen receptor, AR, and progestin receptor, PR, typically bind as homodimers, meaning two identical receptor units come together to inverted repeat sequences on the DNA.
Then you have the fascinating group of other hormone receptors and metabolite ligand receptors, such as the thyroid receptor, TR, retinoic acid receptor, RAR, vitamin D receptor, VDR, and the peroxisome proliferator activated receptors, PPAR, ERA, INGA, along with FXR, LXR, PXR, and SAR. These are particularly interesting because they bind as heterodimers, always with RXR, retinoid X receptor, as a common partner, to direct repeat sequences. Table 42-5 provides compelling examples of specific ligands for these receptors, for instance, fatty acids for PPARs, bile acids for FXR, oxysterols for LXR, and even xenobiotics, foreign compounds like drugs or environmental toxins for CRR and PXR, and the diverse processes they affect. This truly highlights the body's internal sensor system for not just endogenous hormones, but also for dietary components and external chemical agents. And then, as we mentioned, there's a group of orphan receptors for which no ligand is yet known, but which bind as homodimers or monomers to direct repeats, holding tantalizing secrets for future discovery.
The discovery of this nuclear receptor superfamily has led to a profoundly important understanding for anyone interested in biomedical science. It precisely explains how an incredibly wide variety of metabolites, which are products of your own body's chemistry and xenobiotics, those foreign compounds like drugs and environmental toxins regulate gene expression. This impacts everything from your metabolism to how your body detoxifies and eliminates both its own normal byproducts and exogenous agents. Not surprisingly, this entire field, this sophisticated sensor system, is a fertile ground for the investigation and development of new therapeutic interventions, precisely because these pathways are so central to maintaining health and correcting disease states.
To truly complete the picture of nuclear receptor action and achieve a professional level understanding, we must delve into the large number of nuclear receptor co-regulators that also participate in regulating transcription. These aren't just passive helper proteins. They perform crucial active functions like chromatin remodeling, a process that involves modifying histones, influencing DNA methylation, and repositioning nucleosomes, all to make DNA more accessible for transcription. They also modify transcription factors themselves through various enzymatic activities, and critically, they act as the essential communication bridge between the nuclear receptors and the basal transcription apparatus, which is the core machinery that starts gene transcription.
The sheer number of these co-regulator molecules is truly astounding, exceeding 100 known molecules, not even counting species variations and splice variants that add even more diversity. The very first of these to be thoroughly described was the CREB binding protein, or CBP, and its close relative, P300. CBP binds to a specific phosphorylated serine residue on CREB serine 137 and mediates transactivation in response to CAMP signals, effectively acting as a coactivator. Importantly, CBP P300 interact directly or indirectly with a multitude of different DNA-binding transcription factors, including AP1, STATs, nuclear receptors, and CREB, as intricately shown in Figure 4213. They also bind to the P160 family of coactivators, a protein kinase known as P90, and even RNA helicase A, showing their central role in the transcriptional machinery.
This is truly crucial for anyone seeking deep insight. CBP-P300 possess intrinsic histone acetyltransferase, or HAT, activity. This means they are enzymes capable of adding acetyl groups to histones. Why is this important? Because acetylating histones facilitates the remodeling of chromatin, essentially loosening the tightly packed DNA structure into a more open, transcription-efficient environment, making genes more accessible to be read. Histone acetylation and deacetylation play a critical, dynamic role in controlling gene expression by dictating chromatin accessibility. And it's not just histones. CBP-P300 can also acetylate non-histone protein substrates, like various DNA-binding transcription activators and other coregulators, further impacting overall regulatory responses. Their multifaceted enzymatic activity is a key aspect of their power.
Another major and important family of this coregulator network is the P160 family of coactivators, all named because they are approximately 160 kilodaltons in size. Examples include SRC1, ENTO-A1, GRIP1, TIF2, and SAU2, and many others, as listed in Table 42-6. They share several common properties crucial for their function. They specifically bind nuclear receptors in an agonist and AF2 domain-dependent manner, meaning their binding is triggered by an activated receptor. They possess conserved amino-terminal basic helix loop helix, BHLH motifs, a weak carboxyl terminal transactivation domain, a strong amino-terminal transactivation domain, and crucially, at least 3 of the LXXLL motifs, which are specific amino acid sequences required for critical protein-protein interactions with other coactivators. And like CBP-P300, they often possess intrinsic 8 activity. Mutations in this 8 domain severely disable many of these factors, powerfully underscoring the critical role that histone acetylation deacetylation plays in controlling gene expression.
On the flip side, balancing the activators, we have the corepressor family, including important members like NCR and SMRT. As we saw earlier with thyroid hormone receptors, these corepressors function to tonically repress gene transcription in the absence of ligand, keeping genes quiet until needed. And then there are the mediator subunits such as TRAPs, DRPs, and ARC, also listed in Table 42-6. These proteins represent subunits of the much larger mediator complex, which acts as a crucial molecular bridge, directly linking the nuclear receptor coactivator complexes to RNA polymerase II and the entire basal transcription apparatus, thereby directly facilitating the initiation of transcription.
The exact and dynamic roles of all these coactivators and corepressors are under intensive investigation, because many of them have not only structural roles, but also intrinsic enzymatic activities, leading to precise modifications like acetylation, phosphorylation, methylation, sumoylation, ubiquitination, proteolysis, and cellular translocation. All of these modifications can profoundly alter their activity and their specific targets. If we truly connect this to the bigger picture, it's not a simple static one-to-one interaction. Certain specific combinations of coregulators are responsible for particular ligand-induced actions through various receptors. And these interactions on a given gene promoter are incredibly dynamic, constantly assembling and disassembling. Scientists have observed complexes of over 45 transcription factors simultaneously working on a single gene. This is truly the ultimate orchestration of gene expression, where countless factors are interacting in precise, dynamic, and integrated ways to achieve specific cellular responses that define life itself. So what does this all mean? We've taken an incredible deep journey from a simple external stimulus to the intricate ballet of molecules inside your cells, ultimately leading to precise changes in protein activity and gene expression. We've seen how hormones, cytokines, and growth factors are absolutely central to your body's remarkable ability to adapt, maintain balance, and even fight disease.
To recap some of the key insights for achieving mastery, the elegance of signal amplification, where even a single hormone receptor binding event can generate a massive cascade of intracellular signals, is truly remarkable. We've explored the distinct yet often interconnected pathways for group I hormones, which utilize intracellular receptors and directly regulate genes in the nucleus, and group 2 hormones, which bind to membrane receptors and generate diverse second messengers.
And the crucial role of those second messengers, CAMP, cGMP, CA2+, and phosphatidylanides, in diversifying and propagating signals throughout the cell, affecting a wide array of biological processes from muscle contraction to enzyme activation, is undeniable. We also dove into the profound complexity and surgical precision of kinase cascades, such as the insulin, JAK-STAT, and NFD pathways, and how they integrate multiple signals, allowing for nuanced, coordinated cellular responses.
Finally, we've gained deep insight into the vast nuclear receptor superfamily and its intricate array of coregulators, illustrating the incredibly dynamic, fine-tuned, and multi-layered control of gene transcription. This level of control is so sophisticated that it can even be influenced by seemingly disparate factors like components in your diet and the very drugs we use as medicines.
This level of intricate control, with so many feedback loops and crosstalk points, makes you wonder, given the sheer overwhelming complexity of these pathways and their interconnectedness, how does the body ever get it right? And when it doesn't, when these pathways malfunction and lead to disease, how precisely can we intervene to correct it? What stands out to you, our listener, as the most surprising or elegantly designed part of this molecular machinery? This fundamental knowledge truly underpins so much of modern medicine, from treating chronic conditions like diabetes to inflammatory diseases, and it continues to inspire new therapeutic breakthroughs. And that, dear listener, is the essence of today's deep dive into hormone action and signal transduction.